My name is Iain Comerford, and I am at the University of Adelaide. Well, you can probably tell from my accent that I'm not from Australia. I'm originally from Glasgow in Scotland. I've been in Australia for 17 years. Outside of work, my main hobby is running. When I was growing up in Scotland, I knew a number of people who suffered from MS. Some of them were quite close to me. And when I was studying immunology at university, it seemed to me that the science of immunology had the great potential to have a big impact on improving MS treatments. So that was something that I felt I could make an important contribution to. And ever since my PhD, that's always been in there. There have been lots of really exciting developments over the last 20 years. The improvements in the disease-modifying therapies has been quite remarkable. But there's still a long way to go until we can cure MS. The recent work showing that MS its association with the EBV virus is really interesting, because that suggests that potentially we can control MS much more effectively in the future potentially by controlling EBV infections. Not that that's an easy thing to do. And some of the drugs that have been developed in the last decade or so that target immune cell migration into the nervous system are really exciting from my perspective, because that really proves that we can target that process and have that be beneficial in MS. So really my research is trying to build on that to try and come up with better treatments that can more selectively block the migration of the cells that are really causing the disease, rather than all immune cells. Yeah, so the current project is trying to understand what the different combinations of molecules that are used by the cells that are the most inflammatory cells that get into the brain during an autoimmune disease like MS. So we know that there are molecules that are important for their migration, but it's very complicated. There are lots of different types of these molecules, and we don't really understand which ones are used particularly by the pathogenic or inflammatory cells to get in there. And we think that it's not just one specific molecule, that we can really come up with combinations of molecules that can be effectively blocked or targeted to prevent the migration of the pathogenic cells into the brain. So that's really what we're trying to untangle, is which different combinations of molecules would be the most effective targets for drugs in future. Current treatments for MS are, good as far as they go, but they're not without side effects, and they're not completely effective. So what we hope is that we can improve on those therapies and come up with new drugs in future that can target molecules that are more selectively going to block inflammation without compromising normal immune system function. So it's really about building a better arsenal of drugs that can more effectively treat MS in future. That's really the importance of it. I love working in the lab because I love interacting with the other scientists there and the other students. It's a really stimulating environment. There's always interesting data being produced that stimulates interesting discussion and questions. There's always something new happening on a day-to-day basis, which is really inspiring. There's lots of challenges too, and science isn't easy. We have to overcome lots of obstacles on a daily basis. It is not an easy life in some ways, and funding is not easy to come by, so I'm really grateful for this project grant from MS Australia, because without funding we can't continue our research. So so funding is a major one, and also the time-consuming nature of a lot of the research that we do, and the difficulties that we can encounter when things aren't working and we have to work out why.